Hi to everybody. Today we are going with the next lecture, that is conversion of muscle to meat. So before going to today's lecture, I request all of you to go through the muscle structures which was dealt in the last four lectures, I can say. Totally we have talked about in detail about the muscle structures. Only when you know the muscle structures, you can understand or the importance of the conversion of muscle to meat will be easy for you to understand. And you should also know how the muscle contraction and relaxation is taking place. Already you have studied this in physiology, biochemistry and pharmacology, I hope. So I'm just going to brush up what you have learned in the physiology, biochemistry and pharmacology. This is just revising what I told in the last four lectures about the muscle structure. So each muscle is covered by each muscle is made up of muscle fibers. So each muscle fibers is made up of myofibrils, and each myofibrils is made up of myofilaments, that is the actin and the myosin. Okay. And the whole muscle is surrounded by or covered by the epimysium, and each muscle will contain large number of fasciculae, so which is surrounded by the perimysium. So this is the perimysium, and each muscle fiber or the muscle cell is surrounded by the endomysium. So inside the endomysium is the sarcolemma. So let's understand what is neuromuscular junctions. So this is the junction or where axon of the nerves come and binds with the binds with the muscle. So that junction is called as a neuromuscular junction. The junction that is formed between the axon end of the nerve and the muscle is called as a neuromuscular junction. So this is the axon and this is the tail region where the terminal will come and binds with the or attached with the muscle. Okay, so this is the neuron and this is the muscle. So this junction is called as a neuromuscular junction. For the muscle to contract and relax, lot of changes that will biochemical changes will be taking place in this neuromuscular junction, which we are going to discuss in the coming slides. So you have to understand, so this is the neuromuscular junction, we can say. So here this is called as a synaptic cleft or synaptic cleft, we can say. This is called as where the junction is called as a synaptic cleft, and it has presynaptic membrane. So this is called as the presynaptic membrane. Okay, and this is the postsynaptic membrane. So you can see this is the postsynaptic membrane. So at the terminal ends there are large number of vesicles called as synaptic vesicles. So these are the synaptic, this round things you can see, this called as a synaptic vesicles. And the synaptic vesicles will be containing large number of the neurotransmitter that is called as acetylcholine. So each synaptic vesicles will be containing the neurotransmitter that is the acetylcholine, you can see. And when the nervous impulse or the nerve stimulation comes at the terminal, axon terminal, the acetylcholine will be released from the synaptic vesicles. So this acetylcholine will be released. So you can see here the acetylcholine, this small pink color dot, that is it's symbolic the acetylcholine. So this acetylcholine will go and bind with the receptors. So in the muscle there's the receptors is called as nicotinic receptor or NM receptors. Okay, so this blue color, this is called as a nicotinic receptors, or it is also called as acetylcholine receptors. So once it comes and bind with this receptor, this the neurotransmitters comes and bind with the acetylcholine with this neuroreceptors, what happens is this the ion channels. Okay, these receptors are also called as the ligand gated ion channels, we can say. Okay, let's see here. So this is once the acetylcholine. So this is called as acetylcholine. When this this is in the relaxed stage. Okay, this is in the relaxed stage. Acetylcholine base level always will be there. When this acetylcholine come and binds with the receptor. So this is the receptor that is present in the postsynaptic. Okay, you can see here, this is the postsynaptic membranes. This receptor is present. Okay, this is what represented here, the red color. Okay, in the red color. So this acetylcholine, when it comes and bind with this nicotinic receptors, what happens is the ion channels opens. So once the ion, here you can see in the relaxed state it's closed. When the nerve impulse comes, the ion channels will be opened. Once it opens, what happens is the sodium ion rushes inside and the potassium ions will be going out. So this will lead to what is called as the depolarization. Okay, this is called as depolarization. Why this depolarization is taking place? Because the number of sodium ion that is getting inside will be more when we compare to that of the potassium ion that is going outside. So naturally there is a gradient will be there, or the action potential will be created. This action potential is a self-propagating, it will be a self-propagating. That's what we find here. You can see here, it will be a self-propagating. Once the action potential is created, it will be self-propagating and it keeps on moving. So where it moves to the T tubules. We know the T tubules, we have studied in the previous lecture, the T tubules. So in the T tubules, it will be just passing and it will be going to what you call the sarcoplasmic reticulum. So what is the function of sarcoplasmic reticulum? You know, already we have discussed that in the previous lecture. The sarcoplasmic reticulum is the place where the calcium ion is stored. So we can see, you can see here, through the T tubules, the action potential will be coming, and once it comes to the sarcoplasmic reticulum, what happens is the permeability for the calcium will be increased and the calcium ions will be rushed into, or it will be moving into the sarcoplasm. So normally when it is in the resting stage, the calcium ions concentration in the sarcoplasm is around 10 to the power of -7. Once the calcium ion is released, the calcium ion concentration increase in the sarcoplasm, and when the concentration go up to 10 to 100-fold increase. What do you mean by 10 to 100-fold? That is from 10 to the power of -7, it will be increased to 10 to the power of -6 or -5. At that time, what happens is, so you can see here, at that time what happens is the structural changes. So once the calcium ion comes, so it goes and binds with the troponin C. So it will be binding with the troponin C. So this is the troponin, you know, ITC, we have studied all these things. So it binds with the troponin C. Once it binds with the troponin C, the structural changes will be taking place in the troponin. Due to the structural changes, what happens is the tropomyosin. So in the relaxed state, the tropomyosin will be binding with the site on the actin where myosin comes and bind. So once the structural changes take place, what happens is tropomyosin move away from this site. See, this is the site where the myosin head will come and bind. In the relaxed state, it will be covered by the tropomyosin. But during the contraction, or for the contraction to take place, this will be migrated, or it will be moving, or it will be moving away from this site. So this site where the myosin come and bind is free now for the myosin head to come and bind. So this is what we can see here, the neuroaxon terminal. This is a synaptic cleft, we can say. So the nerve impulse runs down to the synaptic cleft. Once it comes to the synaptic cleft, what happens is the synaptic vesicles will be releasing the acetylcholine So once the acetylcholine is released, what happens is it goes and binds with the receptors that is present in the postsynaptic membrane. So that is a nicotinic receptors that is commonly found in the muscles, and once it binds, the ion channels will open. So once the ion channels is open, what happens is the action potential will be created, and the action potential being self-propagating, it keeps on moving to the T tubules. So the action potential is propagated along the sarcolemma and down the T tubule. So it will be propagated and it will be going to the T tubules, and it will be reaching the triads or the sarcoplasmic reticulum. So once it reaches the action potential reaches the sarcoplasmic reticulum, the calcium ion is released. So once the calcium ion is released, that is around 10 to 100-fold increase in the calcium ion will be taking place in the sarcoplasm. So once the calcium ion is released, it goes and binds with the troponin C. Once it binds with the troponin C, the structural changes in the troponin will be taking place. Due to the structural change in the troponin, the tropomyosin move away from the myosin binding site that is present in the actin. So now the site is very free for the myosin head to go and bind with the actin. So this is what happening in the muscle contraction. You'll be understanding better, and I am explaining here. So what happens is, so this is the, you can see here, you can see this is the tropomyosin. Okay, this is the tropomyosin. You can see this is the myosin head. So for this to take place, again it requires the ATP. So this is one place for the muscle contraction to take place. The ATP is required. So once the ATP comes and bind with this, it will be in the relaxed state, and the myosin ATPase. So there will be a myosin ATPase. So this is, you can see this is the relaxed state. Okay, this is in the relaxed state. You can see the site is bound by the tropomyosin. Okay, so the myosin head cannot go and bind. Once the calcium ion concentration increases, so 10 to 100-fold increases, that is from 10 to the power of -7, it comes to 10 to the power of -6 or -5. When it increases, the structural changes in the troponin will be taking place, and this will lead to the tropomyosin to move away from this binding site. So it moves away, and now it is free for the myosin head to go and bind with the actin. So actinomyosin cross linkage will be taking place. So you can see a contraction phase. In the nutshell, we can say the resting phase, the motor and nerve actions potential arrive at the motor end plates. When it's arrive at the motor end plate, acetylcholine will be released, and it get depolarized because the ion channels will be open. The sodium flux into the muscle fibers or the sarcoplasm. So once the action potential is transmitted through the T tubules to the sarcoplasmic reticulum, the calcium ion is released from the sarcoplasmic reticulum terminal cisternae into the sarcoplasm. So when the calcium ion concentration increase from 10 to 100 folds increase will be there, and it will go and bind to the troponin C. Once it binds with the troponin C, the tropomyosin will be moving away from the myosin binding site that is present on the actin. So for it going and binding, the ATP is required. So the ATP will be going and binding with the myosin head. So we'll be seeing that during a small clipping. I'll be showing that one. Once the ATP binds with the myosin ATPase. So this is the enzyme that will hydrolyze the ATP into ADP and the inorganic phosphates. So the ATP will be hydrolyzed into the inorganic phosphate and the ADP. Okay. So you can see here what is happening during the relaxation state. So when there is no nerve impulse is not coming out during the relaxation state, what happens is the choline esterase will be released. Okay, the choline esterase will be released, So this choline esterase will be breaking down the neurotransmitter, that is acetylcholine. So once the neurotransmitter is broken down, what happens is the ion channel will be closed and the sodium will be coming out and the potassium will be getting in. So that leads to what you call as a re-polarization stage. Okay, repolarized stage you can see here. And once the so again what is required is the calcium should be pumped back into the sarcoplasmic reticulum will be taking place. So the calcium from 10 to the power of minus 6 or 10 to the power of minus 7, the calcium will be pumped back to the sarcoplasmic reticulum. So for that again the ATP is required, which I will be discussing later. And once the calcium is coming inside, calcium is reabsorbed, the relaxation will be taking place because the troponin and the myosin will move away from the actin. So actin myosin cross bridge will be terminated and the troponin myosin will come and bind with the actin. The site will be covered and there will be and there is another thing is that the magnesium will be forming complex, a magnesium ATP complex will be formed. The magnesium will be binding with the ATP and the passive sliding of the filament will be taking place and the sarcomere return to the resting state. This is what happens. So you can see here during the muscle contractions, already I told you the ATP will be coming, the myosin ATPase. The myosin ATPase, that is the myosin ATPase is present in the head of the myosin, which will be hydrolyzing the ATP into ADP and the inorganic phosphates. And the stage 1, what happens is the inorganic phosphates will be released. In the stage 2, the ADP will be released. ADP and inorganic phosphates will be released. In stage 2, the ADP will be released and the energy will be there. So because of that energy, this is called as a power stroke. This stage is called as a power stroke. The myosin will be moving the actin towards the center. So there will be shortening of the sarcomere. So what is sarcomere? The contractile unit of the muscle fiber is called as the sarcomere. So when the sarcomere come or the two Z line come closes, there will be contractions. Again, the new ATP will be coming. So this myosin head is low in energy configurations. So the new ATP will be coming. Again, as the ATP is split into ADP plus inorganic phosphates. So here you can see the hydrolysis will be taking place. And again, there will be energy released will be there. This because of the energy, again, it will go and bind with the actin and it will be pushing towards the center. So the sarcomere length will decrease. So there will be repeated myosin will be going and binding with actin. So in this process will be continuing till the actin till the ATP is there. So for the calcium to pump back into the sarcoplasmic reticulum, that is, then only the relaxation state can take place. For that, there's a mechanism called as calcium pump is working. So what is for the calcium pump to work? That is, it has to pump the or reabsorb the calcium back into the sarcoplasmic reticulum against the concentration gradient. So some energy is required. For that also, the ATP is required. Okay. For the relaxation, so during the muscle rigor mortis or the thing, ATP is mainly used for the relaxation stage, okay, than for the contraction. The ATP is used for the relaxation. In the beginning, to maintain the homeostasis and other things, the ATP will be used. So let's see what is happening. For the relaxation of the skeletal muscle, calcium should accumulate in the sarcoplasmic reticulum. That is, the calcium should move back into the sarcoplasmic reticulum from the sarcoplasma fluid. Okay, the calcium should be pumped from the sarcoplasm fluid into the sarcoplasmic reticulum. That is the concentration. That's what I told. The concentration of the calcium should reduce from 10 to the power of minus 6 or 10 to the power of minus 7 to the 10 to the power of minus 5 to 10 to the power of minus 7. And this take place against the concentration gradient. So by anything, if you want to move against the concentration gradient, we need energy for that. So that energy is for moving of that also we require the ATP. So this is done by a mechanism called as calcium pump system. You must have studied in the physiology this. Here you can see here. So the close muscle relaxation, the calcium ion are pumped into the store. Where is the store? Store is nothing but the sarcoplasmic reticulum, allowing the muscle filament to relax. So an active pumping system requires energy in order to overcome the concentration gradient. That's what I told. Outside it will be more, so it has to be pumped back into the same inside the sarcoplasmic reticulum. So naturally it requires some energy. The energy is provided by the enzymatic hydrolysis of the ATP to ADP plus inorganic phosphates. Okay. The enzymes that is responsible for the ATP hydrolysis is not the myosin ATPase. So in the case of we have seen during for the muscle contraction, the myosin ATPase relax. Here what is involved is the calcium ATPase. So in the sarcoplasmic reticulum membrane, there is a calcium ATPase is present. So this calcium ATPase, what happens is it will hydrolyze the ATP and it will release the energy. Because of that energy only, the calcium is pumped back into the sarcoplasmic reticulum. So once the calcium ion concentration decrease in the sarcoplasm from the 10 to the power of minus 5 or minus 6 to 10 to the power of minus 7, what happens is the structural change, that is reverse what happens during the contractions, it will be taking place here during the relaxation. The structural changes in the tropomyosin will be taking place, in the troponin will be taking place. So the tropomyosin will come back and again bind the site on the actin. So naturally the myosin will be moving and it will be leading to the relaxed state. This is what happening during the muscle relaxation. So you can see here calcium pump transports calcium ion back to the sarcoplasmic reticulum. So it will be, so these are the sarcoplasmic reticulum. You can see it will be pumped back, and as calcium decreases in the sarcoplasm, so as the calcium decreases, what happens is the linkage ceases. That is, actin myosin bond will start breaking, or the linkage will stops, and the muscle comes to the relaxed states. Understood? So this is what happening. Thus, the release of calcium from the terminal cisternae. What is terminal cisternae? You know that sarcoplasmic reticulum and the T tubules together is called as terminal cisternae, or the triads, has twofold effect. It activates the contraction machinery to produce the concentration. So for the muscle contraction to take place, again it will be pumping the calcium. For the muscle to relax, it has to reabsorb the calcium back into the sarcoplasmic reticulum. That is what we call as a twofold effect. Okay, this is called as a twofold effect, we can say. So simultaneously it will be acting. So here you can see in the nutshell what has happened during the muscle contraction and relaxation. We can see when the signal arrive at the neuromuscular junction, from the synaptic vesicle, the acetylcholine will be released. Then this acetylcholine will be binding with the this acetylcholine is nothing but the neurotransmitters. It will be binding with the receptors that is present in the postsynaptic membrane. So that is with the nicotinic receptors, we can say, in the case of muscles. And once it binds, the ion channels will be open. So once the ion channel is open, what happens is the sodium gets inside and the potassium go outside. Since the number of sodium ion coming inside is more and potassium going outside is comparatively less, and action potential will be generated. The action potential being self-propagating, it keeps on moving. From the sarcolemma, it will be moving through the T tubules. Through the T tubules, it will be reaching the sarcoplasmic reticulum. Once it reaches the sarcoplasmic reticulum, the calcium will be released. So the calcium will be moving and it goes and binds with the troponin C. Once it binds with the troponin C, the configurational changes will be taking place, and as the configuration changes take place, the tropomyosin will move away from the myosin binding site that is present on the actin. So the myosin will come and bind with the actin. For the myosin to bind with the actin, it requires ATP. So the ATP will be coming, and the myosin ATPase So remember that myosin ATPase is the enzymes that hydrolyze the ATP into ADP and the inorganic phosphates. So, and the energy will be released. This energy is used for the power stroke. So this will go and bind with the actin, and it will be moving the actin towards the center. So the Z lines comes closer. So when the two Z lines it comes closer, there is a muscle contraction will be taking place. So during the relaxation, the reverse of this will be taking place. So here the re-polarization will be taking place. During the relaxation state, the calcium is reabsorbed into the sarcoplasmic reticulum. So for this reabsorptions, again we require the ATP. So this ATP is hydrolyzed by the ATP that is present in the sarcoplasmic reticulum, that is called as calcium ATPase. So the calcium ATPase will hydrolyze the ATP into the ADP plus the inorganic phosphates, and that energy is used to pumping the calcium from the sarcoplasm to the sarcoplasmic reticulum against the concentration gradient. So once the calcium concentration decreases, what happens is the troponin, when the troponin release the calcium, what happens is again the structural changes will be taking place. The tropomyosin will again come and binds with the actin site where the myosin will bind. So that site will be closed, and the myosin will be going back to its normal stage. Okay, normal condition, that is a relaxed state. So this is what happening during the muscle contraction. So let's see clippings on the cycle of muscle contractions. For the muscle to contract, there are a lot of biochemical changes will be taking place. So let's see how the cross bridges are formed. So already we have discussed during the structure. This is called as a Z lines. Okay, so this is from here to here. It is called as a contractile unit. That's the sarcomere. So this is the red color is the thick filament, and this yellow is a thin filament. And this is the Z line. This is one Z line, and this is one Z line. During the muscle contraction, myosin head will be pushing the actin towards the center. So the sarcomere length decreases. So here you can see the calcium is coming. So once the calcium comes and binds with the troponin C, you can see this is troponin AITC. When the calcium is binds with the troponin C, you can see this is a tropomyosin. Now the site is not exposed. So see, now it is exposed. Once the calcium is bound, so 10 to 100 force increase in the calcium is there, the site will be exposed. And you can see the myosin head is going and binding with the actin. Okay, so the cross bridge is formed. So now the new ATP is coming. When the new ATP comes, what happens is the myosin ATPase that is present in the myosin head will be hydrolyzing the ATP into ADP and the inorganic phosphates. Step 1 is the cross bridge formation. So this goes and bind. And the inorganic phosphate is released. The inorganic phosphate is released. The step 2 is called as a power stroke. So the ADP is released, and there's a formation of there is a energy is there, which because of that energy it goes and bind with the actin and push the actin towards the center. Now again the new ATP comes. When the new ATP comes, the cross bridge detach, and the calcium ion also, so it comes here. You can see this new ATP has come. So again it will be hydrolyzed, and you form ADP and the inorganic phosphates. See, this ATP is coming, calcium ion is coming. So this process will be keep on going. Okay, it will be moved. So there will be actions and relaxation phase. Again you can see during the relaxations what is happening. We can see this is calcium ion concentration comes down, so this site is bounded. So this is what happening during the relaxation stage. So now the muscle. So thank you for watching. Take care. God bless you all. So in the coming lectures, it will be very interesting to learn the conversion of muscle to meat. Once again, I request you all to study, to go through all the 4 lectures of the muscle structure and the muscle contraction also, so that it will be very useful for our coming lectures. Thank you.